All right, so we finished talking about the structure of DNA and how it's double-stranded and has this double helix, and we talked about major groove and minor groove and how these things base pair together. Something to consider here is that these base pairs are fairly flat in nature, or these nitrogenous bases are flat in nature, and if you look at how they hydrogen bond with each other, they base pair in a flat orientation. We'll come back and see this when we look at the actual structure. But let's move on and talk briefly about RNA. RNA, in contrast to DNA, is single-stranded. This doesn't mean that you don't have base pairing. You can essentially have one long strand that curves back on itself and has base pairing involved. In RNA, the thymine is replaced by uracil, and so we have AU pairs instead of AT pairs. Now, while DNA is always a double helix, RNA is more variable in nature, and there's three main types of RNA. So you have messenger RNA, which is transmitting a message and tends to be fairly linear in nature. It doesn't have much base pairing in it. Transfer RNAs, which facilitate the interaction between that message and an amino acid, has a much more defined structure. And I'll show you that in a moment here. So this is an actual transfer RNA and how it looks. There's also something called ribosomal RNA, and that is the RNA that is present in a ribosome. That is what houses both the messenger and these transfer RNAs, and is the actual machine behind putting the protein together. And we'll go into that in later chapters. But as a rough schematic, let's take a look at how this looks. So here we have a strand of DNA. You can see that it's double-stranded. It's anti-parallel, and it's complementary. So here we have ACT. This reading backwards is TGA. Right? So if this is what the message that we want to communicate is, we call that the coding strand. And then we will make a messenger RNA through the process of transcription or copying. We will get this messenger RNA. So see here we have A, C, C, U instead of T, G, G, C, 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 C. And we can break this up into regions that won't turn into a protein, called untranslated regions. And then we have codons that actually communicate the information for a specific amino acid. If you look here, this messenger RNA strand would actually be complementary to this template strand. So it was actually this template strand that we built this messenger RNA on. This messenger RNA then gets moved out into the cytoplasm where it encounters a ribosome. That ribosome binds this messenger RNA, and it can also bind these transfer RNAs coupled to amino acids. These transfer RNAs has a anticodon that would be complementary to the codons on the message. So here we have AUG, here we have UAC. So AU, 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 and GCs. So you can see how they would match together in much the same way that this double-stranded DNA goes together. This would bring in methionine with it. The next codon, this transfer RNA, would bring in serine with it. So each anticodon is coupled to a specific amino acid. This is how reading messenger RNA and consequently reading the gene gets translated into an amino acid sequence. And it is the ribosome's job to hold this in place and make sure that the right parts are going in the right slots. So we want to make sure the proper codon is base paired with the proper anticodon and consequently the correct message. Notice that this also creates a 3-member reading frame, right? So if we were to delete this A, then it would start off with UGA, and then it would go CGG, or GCG. And everything would be shifted over by 1. So reading frame is important, and deletions can cause changes in reading frame. Now let's take a look at the actual structure of DNA. First thing you'll notice here is there are 2 backbones present. So deoxyribose phosphate, deoxyribose phosphate. You will see a major groove here and a minor groove here. Within this major groove, you can actually reach and touch the nitrogenous bases. Here you cannot. Notice that these nitrogenous bases are flat and they tend to stack on top of each other. This stacking interaction between these 2 strands is actually what forms the double helix to begin with. So I'll rotate it around here so you can get a better feel for how it looks. Now, notice that these nitrogenous bases are marked in blue. And what I'm then going to do is move on to a space filling model. And almost immediately you will see that that blue region is touchable in this major groove, but not in this minor groove. So, concept 5.5, nucleic acids store, transmit, and help express hereditary information. So we have double-stranded DNA. It can replicate itself and then direct messenger RNA to be produced that travels out into the cytoplasm where it meets a ribosome. Ribosome facilitates individual amino acids coming in the proper order according to this message and being integrated in. Nucleic acids are made up of a sugar, phosphate group, and a nitrogenous base. We have 2 different families of nitrogenous bases. We do have a difference in DNA versus RNA. We also have a difference in sugar. Again, this is a typo here. This should read RNA. It has an OH group at position 2. It is missing in DNA, so deoxy. That's what's missing. These get linked together in a 5' to 3' direction, referring to the number of the sugars, or the number of carbons in the sugars. Carbon 1 is where the nitrogenous base attaches, so we have the next phosphate attaching to carbon 3, and then it would go to 5, 3, and so forth. It forms a double-stranded helix. The strands are anti-parallel in nature, so 5', 3', 5', 3'. A's base pair with T's with 2 hydrogen bonds, C's with G's with 3 hydrogen bonds. RNAs are typically single-stranded, but that doesn't mean they can't have more complex structures. You get a much more variety in overall structure in RNA. It can be just a single strand with very little structure. Here we have a double-stranded piece of DNA. You can see the coding strand, the template strand, which directs the formation of this messenger RNA. That messenger RNA, you can see, is complementary to this and identical to this, with the exception of the whole U and T issue. That would get moved out of the nucleus into the cytoplasm, where it encounters the ribosome, and the ribosome brings in these transfer RNAs, which have a specific anticodon, which matches a codon and is coupled to a specific amino acid. And that is how the DNA can transmit the information on how to put together a particular protein.